All right. Well, welcome. Today we're going to talk about what is definitely a tricky concept, a confusing concept. So I want to, if I can, kind of walk you through it a little bit. And we are going to introduce the concept of meiosis. Now, you've already learned about mitosis, and mitosis is when a cell asexually reproduces, a eukaryotic cell. So a cell that has DNA inside the nucleus has to go through the process of mitosis to reproduce. Today we're going to learn about meiosis, which is a similar process, but it's the process undergone by eukaryotic cells when they make sex cells. So this is a part of the process of sexual reproduction. So let's take a look and see if we can work our way through it. So here we are, we're talking about meiosis. You will sometimes see it pronounced meiosis. I'm not sure which is correct or if either is correct, but I was always taught meiosis, and that's what I am going to say here. And meiosis is about making sex cells. And as you'll recall from the previous lesson, a sex cell is a cell that has haploid DNA, which is to say only one copy of each chromosome. And that makes sense because when you go to make a new organism through sexual reproduction, it wouldn't work very well if they got 2 copies of each chromosome from mom and 2 copies of each chromosome from dad, because then this new offspring would have 4 copies of each chromosome. And you can imagine every generation you would get a doubling of how many chromosomes, and that would become untenable really quickly. So it is important when we make sex cells that they be haploid, that they only contain 1 copy of each gene and 1 copy of each chromosome, so that the eventual offspring produced can get half of its genetics from its mother's sex cells and half from its father's sex cells. And you'll see that this really is just a variation on mitosis. So just like in mitosis, we are going to sort of start at the end of interphase. And at the end of interphase, we have a cell. There's our cell. And inside the cell is a nucleus. I'm going to show the nucleus like that. And it has got diploid chromosomes, which is to say 2 copies of each. Now, in a normal human cell, that would be 46 chromosomes. I'm not going to draw 46 chromosomes here. Instead, I'm going to draw 2, as if a normal human only had 1 chromosome, 2 copies. This is just to make life simple. And let's take a little note of that. At the end of interphase, the DNA has been duplicated. And the cell is diploid. Which for a human means 46 replicated chromosomes. Which actually means, in a way, there's 4 of every gene, because you can see that this has been photocopied. It's been stapled to itself, just like in mitosis. A copy has been made for the production of new cells. So that's where we begin, and from this we are going to enter meiosis. And meiosis is this process that essentially happens twice. It's basically mitosis twice, with a couple small but important changes. So we're going to start here, and just like in mitosis, meiosis starts with prophase. I'm going to remind myself of my color scheme here. So we have the cell, just like that, and we have the nucleus, but the nucleus will be dissolved, right? So if you recall, we have that nucleus. The nucleus will begin to fade away, and eventually the nucleus will be gone. And I'm just going to show it with the barest remnants of a nucleus, just to show that it is essentially dissolved. And we're going to recall that we have the maternal and the paternal copy of our chromosome, both replicated. In other words, this is the one that originally came from its mom and this one from its original dad, but 2 copies of the same chromosome. So just to keep track of what has happened so far, we can say the DNA will coil and supercoil. To form the chromosomes. We can say that the nucleus will dissolve. And if you recall, we said that we get the formation of these structures called the centrioles, and they begin to produce spindle fibers that are going to go out like that. So we can say centrioles and spindle fibers form. And there we are. So this is completely identical to prophase in a normal mitosis, a normal eukaryotic mitotic cell division. One thing that occurs to me, I think I want to color code this a little differently just to make those spindle fibers and the centrioles more distinctly different from the blue chromosome there. I'm going to kind of show it like that in purple. Yeah, I already feel better about that as a color scheme. I'm going to make these kind of light, because that's kind of how they appear if you were to look in a microscope. Yeah, feeling pretty good about that. Okay. There you go. Try not to be intimidated by my, obviously sensational artwork, but there we are. So, prophase 1, just like prophase in a normal cell. Metaphase is just like metaphase in mitosis, which means we are going to again have our cell, no nucleus at this point, and we are going to have the chromosomes, the homologous chromosomes. Remember, these are the same chromosome, and they're both replicated, which means we have 2 copies of it ready to go, because normally it gets split up. And by now, the centrioles are producing spindle fibers, which will connect to the centromeres of both of these. All right, there we are. And so we can make our notes there. We can say chromosomes line up at the equator of the cell. And at the same time the chromosomes are lining up at the equator of the cell, we are going to get the spindle fibers attaching to the chromosomes. Again, this is exactly like mitosis. And now, in mitosis, I might even make a note of that. And now in mitosis, if you recall, what would normally happen is that in anaphase, the next phase, these would be cut in half. And one replicated copy would go here, and one replicated copy would go there to make a new cell. That would be the normal anaphase process in mitosis. But we're going to see that anaphase is where we get our first change. And if you look here, I've actually notated that. in anaphase, what happens in meiosis is that the chromosomes don't split like they normally would in mitosis. And so we get, okay, just checking my color scheme here. So we have our cell, and just like you would expect, from this spindle fiber, it is pulling a chromosome towards it, and this one is pulling a chromosome towards it. But normally, we would expect to have them split and go half here and half there, and then we form a new nucleus in cytokinesis, and bam, we have two identical cells. But that doesn't happen here. Instead, we get this different thing where neither chromosome splits, and each side of the cell kind of wins, this is oversimplifying, but essentially wins a tug of war and takes one of the chromosomes to each side, one of the homologous chromosomes. So here now we have one of the chromosomes still replicated, coming to this side, while another one of the chromosomes still replicates over there. So those chromosomes don't split. But now we have different chromosomes on each side. The DNA on this side is only half of what would normally be in a cell, and it's different than the DNA on that side. We have different copies, so we can say that. So the big thing is the chromosomes do not split. This is the big chromosomes do not split. This is the big change from mitosis. Instead, one copy of each chromosome moves to each pole. And unlike in mitosis, where because they split, they're no longer replicated, these are still replicated. They are still stuck to an exact copy of themselves. So we have over here a chromosome and the exact copy of that chromosome. We have over here a chromosome and the exact copy of that chromosome. But interestingly, this one is different than that one. So we have different chromosomes on opposite sides of the cell, and that is a big difference. So in a human, it would mean that now there are 23 replicated chromosomes over here and 23 replicated chromosomes over there. Obviously, we just drew one. Right? Whereas up here, there were 46 replicated chromosomes in the middle. All right. And then, just like you would expect, we are going to get the formation of a cleavage furrow, and this is going to split into two cells, and of course we get the formation of a new nucleus on each side. And just for color consistency, I did green on the left. And I did blue on the right. There we go. And so at the end of telophase and cytokinesis 1, we get new nuclei form. And the cells split. And what we are left with is we now have two haploid cells with replicated DNA. And what that means is that each of these has 23 chromosomes, or one of each chromosome, but they're replicated. These can't serve as normal somatic cells because normal somatic cells have to have 2 copies of each chromosome. So they're ready to be made into sex cells, but they're not sex cells yet because they're still replicated. These chromosomes still haven't been split apart. And making these split apart so they're not stuck to an exact copy of themselves is going to be the job of meiosis II. So to summarize meiosis I, at the beginning, we have one diploid cell, which means it's got 2 copies of every chromosome, and they're replicated. And at the end, we have 2 haploid cells, which means they have only 1 copy of each gene, or each chromosome, but they are still replicated. So at the beginning, we have one diploid replicated cell. And here we have 2 haploid, but still replicated cells. I'm going to put a couple more spaces there. Give me a chance to fit that. All right. And so now in meiosis II, we go through that process again, and this time we're going to split those so they're no longer replicated, so they can really serve as sex cells. So in prophase II, I want to draw both these cells, and I understand that this might be a bit clunky, but whatever, try and stop me. So remember, coming out of Metaphase I, we had 2 cells, and so now in prophase II, we're going to again get the nucleus dissolving, and we're going to have our chromosomes here, but we should have the formation of the centrioles and the spindle fibers starting to form like that. So we get the nucleus dissolving, and centrioles and spindle fibers form. And keep in mind, if we were talking about a human cell, we would have 23 replicated chromosomes here. So just 1 copy of each chromosome, but they're duplicated. And here we have 23 replicated. So these are the other copies of the chromosomes. But again, they've been photocopied, they're duplicated. And now when we get to Metaphase II, we know what's going to happen to these cells, which is the nucleus is going to be totally gone, and those spindle fibers are going to actually connect to the centromeres of our chromosomes. So now we've got this, and it really is grabbing on. And we've got this, and it really is grabbing on. And this time, I should write that. So spindle fibers connect to chromosomes. And now when we get to anaphase, what normally happens in mitosis, which is the splitting up of those replicated copies, this time it does happen. And so now we are actually pulling half of the chromosome to each side. Which means finally, we are going to get cells that aren't replicated. And they need to not be replicated before they can be workable cells. Because again, you can't every time you make a new cell have it contain twice as much information. And so now we have the chromosomes split. And move to the poles. And now we have 23 and 23, and these are not replicated. And 23 and 23, and these are not replicated. And I'm sure you can now predict during Telophase II and cytokinesis II what is going to happen, but we are going to get the formation of that cleavage furrow here, and the formation of that cleavage furrow there, leading to the formation of 4 unique cells. And new nuclei will form. I should show them like mid-formation here. And by the time we get here, they have formed. And there we go. And we have 4 cells with, for a human, 23 non-replicated chromosomes. In other words, now finally we have 4 sex cells. These are now ready to become sperm cells, egg cells, depending on whether you're talking about a male or a female in humans. Keep in mind that it is not just animals that do this. Plants also undergo this process. Anything that reproduces sexually. Some fungi do this, lots of different things. But if we kind of think of humans as the examples, at the end of this process, these would be ready to become sperm cells in a male or egg cells in a female. Interestingly, this happens in a male throughout his entire life. They're always producing new sperm cells. In females, they produce all the eggs they ever will produce before they're even born. And then this process doesn't really happen inside them. Those eggs are already pre-existent. But that's a different discussion. So just to review, if we were to go from start to finish, just with the super basics, at the very beginning, we have one cell, and it is diploid and replicated. And we'll say that. One cell, diploid, which we're going to take to mean 46 chromosomes, because again, we're kind of talking about humans here, and it's replicated. And if you follow my meiosis 1, at the end of meiosis 1, we have 2 cells now, and they have taken only 1 of each chromosome, and those chromosomes are still replicated because instead of splitting in half, they went 1/2 to one side and 1/2 to the other. So now we have 2 cells. Those cells are haploid, which means instead of having the normal complement of 2 copies of each, we only have 1 copy of each chromosome, so 23 chromosomes, but they're still replicated. And then they can undergo meiosis 2. And at the end of meiosis 2, we have 1, 2, 3, 4 cells, each with its own nucleus. And now that DNA has split. So we get 1 there, 1 there, 1 there, and 1 there. And we have 4 cells. These are haploid, which means we have 23 chromosomes in each if you're a human, but these are not replicated. And these can then become sex cells. So I hope that made some kind of sense. This is a tricky topic, to be sure. It's a confusing one. It gets a little worse because in the next lesson you're going to see that there's a couple things that happen in meiosis to make it even a little bit more tricky and, a little bit more important because, we'll learn in that lesson why it is that you and your siblings are more different than you might expect just based on this process. But here is at least a really broad overview of what happens in meiosis, and the reason it is different, the reason we don't just end up with what normally happens in mitosis, is really all about this change in Anaphase I, where instead of splitting in half each chromosome, in fact, one copy of each chromosome still stuck together, still replicated, goes to each pole. And instead of getting 2 diploid cells as a result, we end up getting 2 haploid cells that are still replicated as a result. This will almost certainly be a little confusing to you. Ask for help if you need it. Take some getting used to, but once you've got it, you'll have it down. All right.